Well, let's go ahead and get started. I think even though we're in a variety of physical locations, we want to acknowledge the Lekwungen peoples on whose traditional territory the University of Victoria stands, and the Songhees, Esquimalt, and Wasanish peoples whose historical relationships with the land and water continue to this day. So I'd like to welcome you all to this Student Award Recognition event. Delighted to see you all and to have you here. These are student awards that are granted through a variety of donations to the Institute of Aging and Lifelong Health. The Institute itself is committed to promoting both basic and applied research to improve the health and quality of life of individuals across the life course. And this is a really wonderful opportunity for us to meet with each other and to listen about some of the recent research activities that some of these awards have made possible by some of our graduate students. So I'd like to get right into it. This is going to be an exciting event for us to exchange and hear from everyone's research here. I'd like to remind people we're also used to Zoom these days, to mute your microphones when you're not speaking. And I'd like to also begin to say a special welcome to a number of our guests, several of which have made donations that have made these awards possible. Moses Mukasa is Development Officer for the Faculty of Social Sciences at UVic. Chris Ferguson, founder of Bridgeable, one of North America's most awarded independent design consultancy companies, and he's also co-founder of Service Design Canada. He's representative for the Ferguson Graduate Research Award in Digital Health. Kathy Nurmi, Senior Manager, Individual Plans, Pacific Blue Cross, and she's here representing the PBC Health Benefits Society Scholarship in Lifelong Health. And Margaret Klatt, President of the UVic Retirees Association, and she is representative of the UVic Retirees Association Award. So I'd like to begin. I'll be just quickly introducing each faculty supervisor, and then they'll introduce their student for the particular award that was received. And I'd like to begin by introducing Dr. Deborah Sheets and the UVic Retirees Association Award. Deborah. Deborah?
Apologies. Stepped away for one moment here. So I'm introducing Matilde Cervantes. Was that what you want me to do? Okay. So Maddie is one of my former graduate students. Maddie's a mestizo woman born in the peninsula of California, Baja California, Mexico. She draws on a strong background in psychology with a focus on health within mental, social, and environmental domains. She earned a bachelor's degree in psychology and a master's degree in psychotherapy. And in 2018, she received a 2-year grant from the Science and Technology Council in Mexico for graduate studies at UVic. She completed her program in SDH in December, and now she's in the interdisciplinary studies, doctoral studies program. And out of breath, I was running. Her research thesis focuses on the well-being of caregivers. She worked with the Voices in Motion choir and did a lot of advanced quantitative analyses with our data, which was an arts-based intervention, a choir called Voices in Motion. So she's an amazingly creative researcher. She's very interested in gerontechnology, mental health, and arts-based types of research. And it's really a pleasure to have the chance to introduce her today. So over to you.
Thank you so much, Dr. Sheets. Thank you everyone for being here. Thank you, Dr. Scott Hofer, and also I want to acknowledge also the presence here of Dr. McDonald, who is also one of my co-supervisor, and his guidance has been very relevant for my master's studies. So I would like to start with my presentation. I will share my screen. There we go. And now I I hope you can see the PowerPoint presentation. Thank you everyone for allowing me to share this study today. The title of my research is Positive Impacts of Participating in Acquired Intervention on the Psychosocial Well-Being of Dementia Caregivers. This study, as Deborah mentioned, has received financial support from the National Council for Science and Technology, specifically for my grad studies, but also the project as Voices in Motion. I want to acknowledge the funding support from the Alzheimer Society Research Program and the Pacific Alzheimer Research Foundation. And as I say, thank you to my co-supervisors, Dr. Deborah Schiff, Stuart Mcdonald, and Andrea Smith from Sociology. The territorial acknowledgement, I think we did it all together, but I want also to acknowledge with respect the traditional lands in which we are studying, playing, learning, and sharing. Also, I would like to mention today that today is my father, will be my father, 93 years old birthday. He will be 93 today, but he passed away in 2009. And so today is a very special day, January 21st, and I want to dedicate this presentation to his memory and also to my, with the infinite love to my mother, who was his dementia caregiver for 8 years. And this is part of my motivation to research this theme as well. As you know, dementia, it's a worldwide concern because actually currently we have 50 million people with dementia, accordingly to the World Health Organization. And the Public Health Agency of Canada reports that 9 persons are diagnosed with dementia every hour in Canada. Compared to other kinds of caregivers, caregivers of persons living with dementia are at higher risk of experiencing social isolation, emotional distress, depressive symptoms, and all of these can adversely affect their psychosocial well-being. And as we know, as the number of people diagnosed with dementia rises, so does the need for more dementia caregivers. So I think that we need to look for solutions and alternatives to support dementia caregivers. There are different ways to support, but I believe that non-pharmacological alternatives are one of those great ideas. And arts-based intervention, specifically acquired intervention, such as Voices in Motion Choir, it's a great alternative, low cost effective and non-pharmacological intervention. I think that singing in a choir, we know because with the literature review and other projects similar to this, singing in a choir and the impact of music is such an important piece. They can increase well-being, increase positive emotions, decrease negative effects and anxiety levels. They can also increase social connections and decrease the stigma of dementia. So the objectives or purposes for this study are to explore the impact of dementia caregivers participating in acquired intervention and to explore the levels of distress and the psychosocial well-being, but not only to explore those, but also to explore the association between them in a time-varying approach, which is across time during the choir seasons. And I did this with a model. Well, I will explain more about the methods later in the next slide, but I wanted to explore the association specifically between social activities and effect and caregiver distress and effect. And effect, I'm referring to positive and negative affect, and I will explain more about that later as well. Also, the psychosocial well-being is a multidimensional construct, and for this study, it has been operationalized as containing three main domains, which are emotional dimension, affective dimension, and social dimension. So with these three main dimensions, I use 3 measures, 3 instruments to measure those 3 components of the psychosocial well-being. And just to reiterate, reiterate the Voices in Motion project. It's an intergenerational choir, a community-based choir for people with dementia and their caregivers. So they had the opportunity to participate, not only the people with dementia, but also family, friends, and caregivers. And the first Voices in Motion choir in Victoria was launched as a pilot project in January 2018. So the data that I use was from 2018 and 2019. And I want to explain here more about the methods. Actually, to go more in detail to that, the participants, they were 34 diets, a caregiver with the person with dementia, but this study is focused only on the caregivers. Yes, so the age mean was 68 years, and 81% were female and 18.8 were male. This is consistent with the literature and with the things that we know already, that usually female are more the majority of people who is in the role of caregiver, right? Then the data were collected using surveys, and this study used the data from the caregiver survey, specifically from 3 sections, as I mentioned it, according to the multi-dimensional model that I share with you. These 3 sections of the survey I use to measure affect, social activities, and caregiver distress using the SARIT burden interview. Information from social activities from the REACH protocol, and the positive and negative affect schedule known as PANAS to measure affect. So for the analysis, this study uses longitudinal data, and a measurement burst design was used. A multi-level coupled model of change was employed to examine 2 variant association with in-person and video in-person. That was part of the purposes that I explained it in the slide that I just passed. So when I'm talking about within person, I will be talking about the individual impact or the intra-individual impact, and between person will be more like a group level, a group impact. And the reason I want to clarify this is because we have a limited time today, so I curated I selected the most relevant graphs to share with you today, with the most that I consider the most relevant findings to talk about positive impacts. And before we go to the findings and to share the graphs, I want also to show you this equation that maybe can be very complex, but because this study is it's with a quantitative approach, we use this equation, and it's a very important one to go deeper in the looking for answers. So it's going to be a key one for the graphs that I'm going to show you today. And just let me move this here. Thank you. So the coupled model of change for the association between caregiver distress and positive affect at the within person level, we have this graph here. And what you can see, it's a graph that is decreasing, right? So what it means that on occasions when caregiver distress is higher, positive affect tends to decrease or diminish. It's a within person level, and it's consistent with the with what we were expecting, right? Because if you have more caregiver distress, positive affect will be diminished. And then for caregiver distress and negative affect, we found the opposite trend, which means that negative affect is increasing by every unit of increase of caregiver distress. If we have more caregiver distress, we have more negative affect. That is what is this graph telling us, and it's between person. So this is a result that was found in a group level. Also talking about another interesting graph, an important one that I found in my study, it's also in the between person or at the group level. And this graph indicates that when social activities increased by 1 unit, there was a corresponding increase as well in positive affect in between person level. This is important because it means that when people was more engaged, or it is suggesting us that when a person was more engaged in a social activity, their positive affect will increase. So the findings overall showcase a significant association between caregiver distress and affect across time. But let's specify which affect are we talking about, right? So importantly, when caregiver distress was higher, positive affect diminished and negative affect increases. Also, it showed, and this is a finding that makes sense, because the expectation is that every time with caregiver distress increase will be an association parallel within the increasing of negative affect. Also, this study showcases an increase of positive affect in association with an increase in engagement in social activities. The choir intervention then may have a positive impact on caregiver psychosocial well-being. The implications of this study advances our understanding of the impact of an arts-based intervention, in this case, the choir intervention known as Voices in Motion, as a support source for dementia caregivers. This research can contribute to knowledge and offer perspectives on how facilitating social engagement, such as the choir intervention, may represent a promising low-cost lifestyle intervention and a non-pharmacological alternative to support the well-being of dementia caregivers. Findings can also inform efforts to develop community arts-based programs to support family caregivers. Thank you so much, and I just want to reiterate that particular attention should be given to high burden caregivers who are vulnerable to the negative effects of dementia caregiver. We should take care of them. We should hence be targeted by psychosocial interventions such as the choir that can help them to cope while fostering and protecting their psychosocial well-being. Thank you so much.
Maddie, thank you so much. Well done. Yeah, we can't hear all the clapping, but I've just seen it. I understand we'll have time for questions at the end. So there they are. It's good to see. Anyway, excellent presentation. Thank you. And thank you, Deborah and Stuart, for supporting Maddie in her work. Really, really great. Next up is Andrea Pitchinen, who will introduce Jamie Knight for the Ferguson Graduate Research Award in Digital Health.
Well, it is my distinct pleasure to introduce Jamie Knight. She's certainly an outstanding graduate student and an outstanding person who has impressed me from the moment I first met her. Her forward-looking and open-minded approach naturally guides her to the cutting edge. In the time since she's been at UVic, and in fact, from what I know before that, she's never been intimidated by what she doesn't know yet and has eagerly applied herself to developing skills in a whole range of domains that have all come together in a really nice way to support her interest in digital health. So for example, she's developed strengths in complex multivariate and longitudinal data analysis, which is sort of a theme here today, it seems like, in the use of functional near-infrared spectroscopy, which relative to MRI has a kind of more mobile component to it. She's learned about olfactory testing, which is quite unusual and on the edge. And at the other end of the spectrum, in conducting focus groups and qualitative analysis, which can provide essential information for understanding both processes at the individual level and also helping us to understand preferences and feasibility for implementing research that participants are going to have to take home with them and make a part of their day-to-day life. So I think the Ferguson Award in Digital Health is definitely appropriate for her, and I would like to thank you for awarding it to her and congratulate her on receiving it. Jane.
Thank you. So that's such a lovely introduction, Andrea. Thank you so much. I will share my screen for all of you. All right, how's that showing up? Okay, whoops. All right, hello everyone. Thank you so much for being here today. And I'm really excited to share with you just a little bit about my research and how we can use digital health innovations in combination with smell, also called olfaction, for the early detection of dementia and cognitive decline. So being mindful that we only have 12 to 14 minutes, I'll just briefly give an overview of research to date and then cover some of my past, current, and upcoming projects. And of course, if you have any questions throughout the presentation, please feel free to post them in the chat as well. My main areas of interest, as Andrea mentioned, are sensory functioning, cognition, and how those interact with personalized medicine and digital health. But at this point, you're probably wondering, why smell exactly? So smells are volatile chemicals that are carried by the air to your nose or through the back of your mouth and up through the back of your throat. The olfactory epithelium, which you can see at the top of the photo on the right-hand side in yellow, reacts to those odor chemicals, and the signals travel from the olfactory bulb to the central limbic parts of the brain. The pathways are well mapped for both smells coming in through the nose as well as in through the mouth, as you can see here in this next photo. So smell, along with taste, comprises our chemical senses. It is a very important and often overlooked part of everyone's lives, and smell is the only sense with cranial nerves that are directly attached to the cerebrum without stopping at the thalamus, sort of a relay station for the other senses. 3 of the main functions of our olfactory system include safety, nutrition, and well-being. So similar to an alarm system, smells alert us to important things in the environment. For example, everyday safety can be impacted when an individual can no longer smell when food has spoiled or if there's a gas leak. Food choices also can suffer, and with a loss of smell, people choose foods that tend to trigger our 5 taste receptors, and those are sweet, salty, sour, bitter, and umami, which is savory, and those you can sort of get a sense that those are really kind of junk foods often, and sometimes people can even add sugar or salt to their foods to boost the flavor as well. And then over time, these poor food choices can lead to other medical conditions such as high blood pressure, kidney disease, or diabetes. And odors also influence many aspects of mood, memory, and emotions. So interestingly, humans can distinguish hormones and even detect genetic complexity in potential partners, meaning smell really supports sustained intimacy in relationships and is also relied on to form and maintain those close personal bonds. And in addition to these very important functions, it can also serve as an indicator of brain health. For example, 2 common neurodegenerative diseases, Alzheimer's and Parkinson's, can be predicted through the loss of the sense of smell. In Alzheimer's disease, 90% of people who have been clinically diagnosed also have anosmia, which is a loss of smell. And research has shown that smell is one of the earliest indicators, and that the degree of smell impairment is also reflected in the severity of AD pathology, of Alzheimer's disease pathology, meaning that people with more pathology at death also had poorer smell scores. And in Parkinson's, an unexplained loss of smell is also associated with a 12.5 increase in the risk of developing Parkinson's within 5 years. And the prevalence of olfactory impairment in Parkinson's disease ranges from around 73 to 90% and increases with the progression of the disease. The underlying reason for these connections between olfaction and neurodegenerative disease lies in the overlap of these brain structures that support smell, memory, and other cognitive functions. So in Alzheimer's disease, pathology progresses in a very predictable way through the brain. And as you can see in this image, the hallmark pathologies are the amyloid plaques and the neurofibrillary tangles. And the first regions affected with Alzheimer's disease pathology include the entorhinal cortex, which you can see highlighted in pink, and it's in both of the first stages of the pathology in Alzheimer's disease. And you can also see it is one of the main olfactory processing areas. So I've highlighted them all there for you to see the locations. After considering previous research and what we know about the length of time it takes for Alzheimer's pathology to build up in the brain before cognitive decline is detectable, it became apparent that the olfactory pathways were actually sensitive enough to detect those earliest damages from the underlying neuropathology of Alzheimer's disease, potentially indicating a risk 10 or 15 years before cognitive decline could be detected clinically. But we really needed more information on what degree were they linked. Did the smell scores decline first and then cognition? How were they linked exactly? Some of my past work looked at how olfactory test scores and cognitive test scores fluctuated together within the same people and provided some evidence that on days when people were performing poorly on smell compared to their own average, they also performed worse than their own average on cognitive scores, meaning that performance was linked such that if one changes, so does the other one. And this idea really pointed us towards the potential that in-depth daily monitoring of these variables could have insights regarding fluctuations in both smell and cognition. We also found that individuals with higher smell scores were less likely to transition to mild cognitive impairment and dementia from a non-impaired state, and they were also living for an estimated 6 years longer free of cognitive impairment. And my current project built off of these findings, and we hope now to clarify how cognition and smell could be used in order to monitor and or evaluate these early stages of dementia. So we're currently looking at how brain activation differs for smell and cognition when people are put under a heavy cognitive load. So we used near-infrared spectroscopy to monitor functional cortical brain activation while participants completed these mobile cognitive assessments, as well as olfactory testing. So once our analysis is complete, we'll have a better idea if these methods are feasible to use in monitoring and evaluating changes in performance, and if we can expand this. For example, if we could expand it using Fmri or PET to determine if early declines are related to the buildup of the Alzheimer's pathology, and if we can use these methods. So having personalized data on cognitive performance will be a key facet in monitoring ability changes in these prodromal early stages of dementia. Currently, there's very little information on what performance looks like in these early stages, and really it's most feasible to have individuals collect this data on their own, similar to how people now monitor their heart and physical activity with wearables like the Apple Watch and the Fitbit trackers. And our lab designed and collected data with older adults in the Victoria area to capture mobile ambulatory assessment of cognition, stress, well-being, and physical activity. We really wanted to know how people are doing in their own environment as compared to like a lab setting, and examine the feasibility of using these mobile cognitive assessments for these early detection of cognitive decline. We also collected data on sleep and blood pressure, and of course, olfaction, and we called this project DASH. And using this DASH data, one of the upcoming projects we have builds off of that previous paper I was talking about, the one that looked at how olfaction and cognition scores change together. And so by examining the extent to which olfaction accounts for variations in this day-to-day ambulatory cognitive tests, so we hope and we thought really that this was going to be able to describe whether lower olfactory scores are predicting a greater variability in the day-to-day cognition of older adults. And perhaps we can use this to determine the efficacy of these tools and increase the specificity of olfaction for the early detection of dementia. Lastly, we're also planning new data collection within our DASH participants to examine whether higher baseline olfactory scores were protective against declines in well-being throughout COVID. And you're going to hear more about this from Nathan Lewis in one of the upcoming presentations. So I'll say no more about that. And in closing, I would love to say thank you so much for listening. And really, the old adage seems to still hold true. It is worth the time to take and stop and smell the roses. Thank you.
Thank you so much, Jamie and Andrea. Really great research. Nice research program, and you've brought a lot together. Thank you. We'll hold again questions till the end. Next up, is Jody Garlick will introduce Colleen Lacey for the Alice Lupoy Graduate Scholarship. Jody.
Thank you. It's my absolute pleasure to introduce Colleen Lacy today. Colleen completed her honors degree in psychology, which involved an advanced neuroimaging project in my lab that investigated depression and Parkinson's disease. And then I was absolutely thrilled when Colleen decided to continue on in my lab. She's currently a master's student in the clinical psychology program, specializing in neuropsychology. And over our time together, I've really appreciated Colleen's leadership and her enthusiasm. She really brings a thoughtful approach to a wide variety of projects in my lab. Colleen's current project focuses on using MRI to better understand healthy aging, and she'll tell you more about that today.
Thank you so much for that warm welcome, Jody. I'll go ahead and share my screen with you guys. All right, so as Jody said, I worked on my honors with her, and I'm excited to be working on my master's with her as well in the Garlick lab. And I would also like to say a big thank you to the donors of Alice Lupoy Award for helping to fund the research. So I'll get into talking a little bit about my research, which is all about ApoE genotype and its impact on brain structure and function in the aging population. So a little bit of background, I think known to most of you folks, Canadians are living longer at an increasing rate, but unfortunately living a longer life does not necessarily equate to living a better quality of life. And a big part of that is neurodegenerative disorders that are rising with the growing population, such as Alzheimer's disease. And so research is really emphasizing early detection, such as biomarkers that might be able to assist in understanding whether or not someone is at risk of developing something like Alzheimer's disease or following a more healthy aging trajectory. And then with that, early detection means earlier intervention for these individuals. So with that in mind, we might ask, what does the brain look like when it's healthy before any signs of cognitive decline come into play? We can study the healthy aging brain to try and understand early onset changes to cognitive function and also to brain structure. And hopefully this will help us understand who might be at risk and why those individuals might be at risk for Alzheimer's disease, and looking at underlying mechanisms in the brain while it's still considered to be relatively healthy. So for my research, the current study is rooted in a theoretical model of cognitive aging called the Scaffolding Theory of Aging and Cognition, or STAC-R for short. So as complex as this theoretical model looks, it's including a lot of different variables that play into part how someone's course of cognitive aging goes. And so I've broken it down into three kind of components here that will provide foundation for my research. So the first is looking at pre-existing factors that might be impacting biological aging trajectories. So factors contributing to neural resource enrichment or protective factors, factors that are contributing to neural resource depletion or risk factors. And then the second part of this model is looking at, well, how do we actually measure these in relation to brain structure and function? And that could be using a variety of different neuroimaging methods, such as MRI. And then finally, how does all of this link to how someone is actually performing at a cognitive level on cognitive measures, and how is that rate of change occurring over time? So to address the first component of this model, I'm looking at how biological genetics might impact aging, and in particular, the apolipoprotein E or ApoE genotype, which many of you may have heard of in the context of ApoE4, as it is a well-established risk factor for Alzheimer's disease, and in particular, late onset Alzheimer's disease. But there's also other alleles that come into play with ApoE, such as the ApoE3 allele, which has been associated with no increase or decreased risk for Alzheimer's disease, as well as the less studied and less well understood E2 allele, which has been associated with mechanisms involved in protecting the brain against Alzheimer's disease. And since each individual has 2 copies of these alleles, you can see that someone's risk might be complicated. For example, if an individual holds 1 copy of the E4 allele and 1 copy of the E2 allele, what does that look like in terms of their risk for later developing something like Alzheimer's disease? The next component is, how does ApoE actually impact the brain, and how do we look at that? So for my research, I'm using MRI techniques, which is a non-invasive and commonly used method to investigate neuroanatomy. So we can look at things like gray matter volume, which is mostly comprised of cell bodies. And we can also look at white matter, which tells us something about the connectivity between these different regions. And so here on the right are some examples from another paper to kind of compare and contrast what an MRI, structural MRI might look like in a healthy brain versus someone who's in the later stages of Alzheimer's disease. And as you can see in the individual on the bottom with Alzheimer's, their brain appears quite shrunken and quite shriveled compared to that healthy brain. But we're wondering, even if we look in individuals with a seemingly normal, healthy brain, are there kind of microstructural changes that are evident even in the earlier stages before someone even gets to these later stages? So there's a body of research that has investigated these different ApoE alleles in relation to cognition and MRI in healthy individuals, so individuals who are not yet cognitively compromised. And research has kind of shown that individuals with one of those E4 alleles or 2 of the E4 alleles are associated with decreased gray matter volume and white matter integrity in temporal and hippocampal regions, areas important for learning and memory, as well as decreased overall cognition and memory and executive functioning, despite still being in that normal cognitive range. But the literature is mixed, so other studies looking at healthy populations haven't seen the same thing. And with the E2 literature, there's been shown some increase in gray matter volume and white matter integrity, so kind of opposing or mirroring the E4 individuals, and as well as increased episodic and executive functioning. But again, the research is mixed, so some individuals or some studies aren't finding these same things. So that really brings me to kind of looking at these research gaps and how the current study will address these gaps. So the current study aims to use multimodal imaging and multi-cognitive domain measures to assess brain structure and function in healthy older adults with the different ApoE genotypes. And it's hypothesized that there will be gray and white matter differences between people, healthy people with these ApoE alleles, as well as slight differences in memory and executive functioning, differences between these ApoE alleles. So the data that I'll be using to investigate this is data retrieved from the ADNI, or the Alzheimer's Disease Neuroimaging Initiative, which is a publicly accessed secondary database that it includes a lot of different cognitive and neuroimaging measures for individual cohorts of healthy cognitive aging all the way down to Alzheimer's disease. So for the sake of my research, I'm looking particularly at healthy individuals who either have E4 alleles and are maybe in a risk group, the ApoE3/E3 control individuals, as well as a group looking at the E2 protective allele. So the individuals in my study so far are in the mid-70s range. The larger groups tends to be the E3 group, as that's more prevalent in the general population relative to E4 and E2. And in order to be qualified as cognitively healthy, individuals need to be falling in that healthy range on measures of memory, as well as not showing any symptoms of dementia. So first things first, I've looked at some of the DTI. So the DTI is measuring the white matter integrity using a measure of water diffusivity in the brain. And so we can use this to tell how intact someone's white matter connections might be. And I can compare that between the different groups. So after pre-processing, results might look something like this, where we can see those white matter tracks and certain highlighted color areas that show any significant differences between the groups. So preliminary results from my study have shown that there are no significant differences in white matter integrity between the three groups, as well as no significant differences in memory or executive functioning scores across these groups. So this suggests that there's comparable cognitive functioning and white matter structural integrity across these groups in the healthy stage. But I have yet to look at brain volume and gray matter. And this is consistent with some previous findings as well. So my next step, of course, will be looking at gray matter and whether or not there will be gray matter volume differences between the groups. And so we would expect my results to come out looking something like this, and then we can tell if there's any significant differences from that metric. And bringing it back to the greater impact, hopefully, this research will help with early biomarker detection and helping to understand underlying mechanisms of ApoE in the healthy brain, and kind of determining what structural changes are or are not evident in the healthy stage of aging. So preliminary results suggest that DTI may not be detectable in the earlier stages of aging associated with ApoE, but potentially gray matter volume would be. And hopefully overall contribute to the general knowledge of healthy aging and abnormal aging in the context of the growing aging population in Canada. So thank you very much for listening to my talk today.
I'll clap for everybody. Thanks so much, Colleen. It was a great presentation. And Jody, thank you. Again, questions towards the end. Next up is Nathan Lewis. And I am delighted to be his advisor and to give a bit of an introduction to Nathan. Nathan has really been a powerhouse. Just he cannot say no. He is on so many excellent projects and contributing so much to each of them. He's working in an area of applying often advanced statistical modeling, including multi-state survival modeling, look at lifelong cognitive engagement and lifelong cognitively stimulating activities and its effect on rate of progression to dementia. He's using change point models to examine the effects of social and cognitive enrichment on delaying cognitive decline, funded by an Alzheimer's Netherlands grant, working on understanding how changes in prevalence of dementia trends can be accounted for historically and geographically. And something he's long been interested in too is purpose in life and its role in cognitive functioning as a protective factor against depression-related cognitive deficits and dementia. He has a number of recent publications, one on purpose in life as a motivational resource for goal-directed behavior, and a couple of other papers, such as within couple longitudinal personality and health behavior dynamics in health psychology. He's also funded by the Alzheimer's Society of Canada Research Program Fellowship and by an Alzheimer's Netherlands International Fellowship, where he's supposed to be in the Netherlands right now. But unfortunately, hopefully, it will happen in the fall or early next year. And with that, Nathan, I invite you to present your current research. Thank you. Oh, and I should have mentioned, Nathan was awarded the Nina Chappell Scholarship. And now you can begin. Thanks, Nathan.
All right, thank you, Scott. Now the pressure's on to live up to that very great introduction. Thank you. All right, so I am going to talk a little bit today about concept of psychological resilience and cognitive reserve and how that relates to some of our ongoing data collection efforts in our lab. So I have, as Scott mentioned, I have a lot of research interests. I have hands in many pies, I guess. Should probably pick a more COVID-friendly metaphor, but lots going on. But today I'm going to talk about three things. So talk about a bit about purpose in life and how that relates to psychological resilience in the face of some common challenges that older adults face. I'm going to talk a little bit about the concept of cognitive reserve and this idea that some people are better able to tolerate neuropathological burden that is typically associated with Alzheimer's disease. And then finally, I'm going to wrap up talking about some different methodologies in terms of design and statistical analyses to help facilitate early detection of cognitive change. So first off, a lot of my past research is focused on this concept of sense of purpose in life, in particular, associations between sense of purpose and cognitive functioning, where this work suggested that purpose predicted better cognitive functioning basically right across the lifespan. But there's also a larger literature suggesting that having a sense of purpose, feeling that your life has direction, is meaningful, and is guided towards some greater life aim, has positive benefits, including reduced risk for dementia, better cardiovascular health, including reduced risk for stroke and heart disease, as well as just generally greater life satisfaction and mental health. So while sense of purpose in life appears to be an important contributor to older adult health, older adults, on the flip side, tend to be more at risk for declines in purpose in life. And people have suggested that this could be in response to challenges such as declining health, as well as changes in typical roles that help give us this feeling of purposefulness. So addressing this question, so I was interested in how do people overcome adversity and common challenges to their purpose and well-being. So one study that came out recently looked at retirement. So for many people, retirement is a good thing. It's a very happy event, but we find that often people do experience declines in sense of purpose in life following retirement as they kind of move away from their work role that may have contributed to their feeling of direction. However, in this study, we found that being active in your leisure time, so being physically active, but also being socially active, doing things like volunteering, keeping mentally engaged, this was associated with kind of maintaining sense of purpose in life across this transition into retirement. And another kind of challenge to one's sense of purpose in life, another study looked at the impact of having a stroke. So people who were recently diagnosed with a stroke often experienced, as you might imagine, kind of loss of sense of purpose in life, especially if they were experiencing physical disability or other lingering symptoms. However, we found that people who perceived greater social support and less strain from their close social relationships, so their spouse and friends and family, they were less likely to experience declines after stroke, and in some cases actually reported feeling more purposeful relative to before the stroke. So I think this research is really relevant in current times as we're all facing these challenges associated with COVID to our everyday activities and the things that we usually feel give us a sense of direction. So I would definitely point towards things like close social connections and staying active in terms of your leisure interests and maintaining these purposeful activities. Another concept that I'm interested in is the idea of cognitive reserve, which really reflects this discrepancy between what we see in the brain versus the actual observable cognitive symptoms. So this research kind of emerged from older findings suggesting that in some cases, over a quarter of older adults who were relatively healthy throughout their life, they didn't show major signs of cognitive impairment. When you actually look at their brains after they passed away, many of them experienced in some cases had very severe levels of neuropathology that you typically associate with Alzheimer's disease, but they didn't express any of the behavioral symptoms. So there's definitely this kind of discrepancy between what we see in the brain versus the actual observable symptoms. So the way I like to think about this is in terms of this figure on the left here. There are two people who have similar brain reserve capacity. I like to think of that in terms of just similar brain size. They have very similar brain anatomy, but the individual on the left is able to tolerate a lot more pathology, as represented by the dash marks, before they actually experience functional impairment. So compared to patient 2, who would show signs of dementia much earlier than patient 1. So importantly, this is not just limited to Alzheimer's disease. Conditions like stroke or traumatic brain injury can also, cognitive reserve is also believed to be involved in how individuals adapt to any kind of brain insult. So importantly, cognitive reserve has implications for the kind of trajectories that we might expect for individuals if they do end up developing dementia. So people higher in cognitive reserve tend to have later onset of cognitive decline, as represented by the point of inflection here. However, this is typically followed by a steeper level of decline, as these people are, I almost like to think of it as like a dam. These individuals are able to hold off the water longer and longer, but once it passes their threshold, there's not much they can do anymore, and they tend to decline at a faster rate. But one of the positive aspects of cognitive reserve in this case is that these individuals often live sometimes years longer without developing any type of cognitive impairment. They don't have the same need of care for as long as someone lower in cognitive reserve. So importantly, and I think this is what shows most promise about the concept of cognitive reserve, is that this is something that's modifiable, seems to be right across the lifespan. So a lot of the research on cognitive reserve is focused on education in early life, but there's also evidence that like mid and late life lifestyle characteristics play a part as well. So if you have work in a cognitively demanding job setting, or in your leisure time you're reading or spending a lot of time mentally stimulated, or have a very active social life, these are things that can help to promote resilience. So it's almost like a cumulative lifestyle factor. And so finally, and this is related to the project I'm going to discuss in a bit more detail, I'm also interested in ways to try to facilitate early detection of cognitive change. So much of the early stages of cognitive decline that I've talked about are typically marked by pretty small changes in cognitive performance. So a lot of the ways that we would typically look at this with longitudinal studies may actually be unequipped to detect changes at such a small scale. Or in some cases, often in clinical studies, they use cutoff points that are based on population norms, which may not account for within person like changes relative to your own personal scores. So if I have a very good memory score, these may not be able to detect that I've declined just because I'm still above a population-based cutoff point. So this is where I think intensive measurement designs, which are also known as burst designs or daily diary designs, which involve a lot of very frequent assessments over a short period of time. This shows promise in identifying some of the more smaller scale day-to-day dynamics of cognition. So for example, variability in cognitive performance is another early indicator of risk for cognitive impairment. So individuals who say within a day, they may score really well on a cognitive test, but then later in the day, they score a lot poorer. This could be signs that they're at greater risk for dementia. So this, as Jamie had mentioned, this slide should look a little familiar. This was kind of our impetus for developing the DASH study, in which we had across a 14-day period where we recruited older adults from the community. We gave them mobile devices and they completed cognitive tasks 4 times a day, as well as measures of mood and stress and all that. And then that was paired with physical data from accelerometers and blood pressure and smell tests as well. So I don't know if you've seen the news, but we're in the middle of a pandemic. So that has added a bit more complexity to all these factors that I've talked about as kind of promoting cognitive health in late life. So in particular for older adults who are at risk for the disease, they also may be more at risk for things like decreased physical activity and social isolation and well-being. These are all things that are importantly linked with cognitive functioning and dementia risk. But unfortunately, the ongoing pandemic may exacerbate some of these issues. So with this in mind, we were hoping to do a follow-up on this DASH study. So the DASH study was originally done in 2019. We're hoping to do an approximately 1-year follow-up on the DASH data, where we'll recruit the same people again, do another burst of data. And we're interested in this. We'll collect quite a bit of data, but we're interested in a few main questions, like, for instance, how has the pandemic impacted the health behavior of older adults, their well-being, and also how this relates to some of these day-to-day dynamics in cognitive functioning. So given that I think a lot of people are stressed out right now, does stress now have a greater impact versus before the pandemic on cognitive performance? So we have a few things going on there, and I'm looking forward to getting into data collection. So thank you for listening.
Nathan, thank you so much. Okay, we hold for applause. And next up, we move to Stuart Mcdonald, who will introduce Cindy McDowell and the recipient of the PBC Health Benefits Society Scholarship in Lifelong Health. Over to you, Stuart.
God Hofer, thanks for turning it over. Great to see so many familiar, smiling faces. Yeah, the sincerest thanks to begin to Scott, Leah, and Joanne for facilitating today's event. It's an absolute great pleasure, as it has been for all of us to this point, to introduce another most deserving awardee, Cindy McDowell. In this case, Cindy is an MSc student in the lifespan stream in the Department of Psychology. And although just in her second year of my growing lab team in a very short period of time, common theme, Cindy has made herself an indispensable team member. She routinely makes impressive contributions to all of her peers' projects, and she finds time to get her own work done as well. Cindy's developing research program has spanned a number of key themes: biological age, functional biomarkers, allostatic load, and ultimately to dementia and lifestyle interventions. And that kind of brings us to today. The research that Cindy's going to overview for us stems from one of our team projects, Voices in Motion. Deborah introduced the project before, and Maddie spoke on it as well. VIM is a study that explores the power of a lifestyle intervention, social singing in this instance, and its potential benefits for improving not only the psychological, but as it also turns out, also the cognitive health of individuals with dementia and their care partners. And as Cindy will speak about a little bit, and I think as she actually wrote about in her application itself, in the midst of a pandemic, Nate, as you kindly reminded us as well, these opportunities for virtual connection are critically important. And as it turns out, one of the things Cindy plans on exploring is to what extent remaining virtually connected might not only mitigate some of the deleterious effects of dementia, but also social isolation more broadly, and what's been termed the socially isolated brain. So Cindy, you're on an impressive trajectory. I just wanted to graciously acknowledge the support from the Pacific Blue Cross Health Benefits Society and this specific scholarship in lifelong health. It will no doubt greatly facilitate Cindy's professional development. So I think on behalf of all faculty and students, sincerest thanks to the PBC Health Benefits Society and to the Institute for making this award possible. Please join me in congratulating Cindy on the receipt of this award and welcoming Cindy's overview for us here this afternoon.
Wow, thank you, Stuart.
Audible, Cindy.
Okay, I am going to go ahead and share screen. Okay, sorry, this never gets any easier. Okay. Okay, yeah, so my research project focuses on the impact of a socio-cognitive lifestyle intervention for persons with dementia. Similar to Maddie, and as Stuart and Deborah discussed, my research is in relation to the Voices in Motion choir, but focusing on the results of the care recipients rather than the caregivers. So as touched on earlier, the Voices in Motion project was created by an amazing interdisciplinary team, including doctors Andre Smith, Deborah Sheets, and Stuart Mcdonald. Voices in Motion, excitingly, is about to gain nonprofit charitable status and is supported by some incredible foundations, including the Alzheimer's Society of Canada and the Pacific Alzheimer's Research Foundation. Before I dive into the current research proposal, I'm going to first provide a bit of a background on Voices in Motion, supplementary to Maddie's presentation, and on the rationale behind these projects. And in doing so, I will actually spend most of this presentation discussing my research leading up to the new proposal. So the aging population and subsequently the number of individuals diagnosed with dementia is increasing rapidly. And as there's no current cure for dementia, individuals and medical practitioners are currently reliant on limited pharmaceutical intervention. Available pharmacological interventions provide mild symptomatic alleviation by masking neuropsychiatric symptoms, but they don't alter or arrest disease progression. Despite pharmaceutical intervention, individuals with dementia often experience a continued deterioration in cognitive function and activities of daily living, in addition to numerous deleterious side effects associated with drug therapy. So lifestyle interventions are therefore becoming increasingly more popular to help mitigate the negative effects associated with dementia, and are being used in tandem with pharmaceutical interventions. Given that dementia can cause feelings of isolation and fear, non-pharmacological interventions, such as art therapies, can be effective by providing a sense of connection and an alternative medium of expression. And music interventions in particular represent a promising approach to improve mood, well-being, overall psychosocial functioning by alleviating negative affect and negative symptoms and improving quality of life. Additionally, music memory is specialized, and brain areas responsible for the music memory system are thought to be relatively spared from Alzheimer's pathology and are typically the last part of the brain to deteriorate. However, majority of music-based intervention studies assess between group differences, typically assessing performance from a single occasion or measure, which makes it difficult to conclude treatment effectiveness within persons. Few studies have examined lifestyle intervention programs for persons with dementia using longitudinal intensive repeated measures designs that reflect real-world intervention engagement. So the Voices in Motion project is an ongoing intergenerational social cognitive lifestyle intervention for persons with dementia and their care partners, as well as local high school students. During the choir seasons, participants attend weekly choir practices, which are followed by periods of socialization. And although an ongoing intervention, data was collected over 3.5 month choral seasons spanning a total of 18 months. A longitudinal intensive repeated measures design, or a measurement burst design, was utilized to collect data on various measures of cognitive, psychological, and physical health at 3 to 4 time points over each season, yielding up to 9 assessments for any given participant. So for my research project, the sample consisted of 33 participants with dementia, and individuals with all classifications of dementia were welcomed and included in the study, although the sample primarily consisted of individuals with Alzheimer's disease. And the primary objective was to examine whether a social cognitive lifestyle intervention can modulate patterns of cognitive change in persons with dementia, and to examine individual differences in rates of change. And the secondary objective was to explore patterns of time-varying covariation between within-person change in global cognitive function and positive and negative affect. So the Mini Mental State Exam, the MMSE, was utilized as a measure of global cognitive function. The MMSE is scored out of 30 with a score of 20 to 24 indicating mild cognitive impairment, 12 to 20 indicating moderate impairment, and less than 12 denoting severe cognitive impairment. And like Maddie's research, the positive and negative affect schedule, the PANIS, was utilized as our measure of psychosocial well-being, or mood. So in relation to the first research objective, results revealed that individuals were declining on their cognitive function at similar minimal rates across the intervention. So to break this down and exemplify these results for everyone, these black lines represent each participant's individual rate of change on their MMSE scores across the 18-month intervention, and the red line displays the average rate of change condensed across all participants. So you'll notice that none of these individual black lines are showing large declines, but rather each individual demonstrated more slow and steady change. Ultimately, participants were changing on cognitive function at similar rates and regardless of their initial cognitive assessment at baseline. And more than that, individuals were remaining relatively stable on their cognitive function and not showing significant declines across the intervention. So specifically, the average rate of decline in global cognitive function was 1.3 units on the MMSE per year, and a total decline of 1.9 units over the course of the 18 months. So to exemplify, an individual who started the intervention with an MMSE score of 20 scored approximately 18 in a year and a half following baseline assessment. This is considerably less than what is expected from a progressive neuropathological disease like dementia, in which the annual decline in cognitive function is expected to be much greater. And to confirm this, we did an extensive search of the literature and found non-intervention studies for those with dementia, and they report on average a cognitive decline between about 2.4 to 3.5 units annually. So the yellow, green, and blue lines here belong to studies with similar samples to ours. They're similar demographics, types of dementia, socioeconomic status. However, the key aspect that differentiate these studies from ours is the choral intervention. So in the absence of an intervention, individuals with dementia have repeatedly been shown to decline well over 2 units on the MMSE, sometimes even up to 5 or 6 units, compared to Voices in Motion participants who were declining by approximately 1.3 units per annum. So the next step was then to examine how or why this might be the case. What specifically about the intervention might protect against this expected severe cognitive decline? Essentially, we wanted to explore whether the intervention might alleviate some comorbidities, which in turn might result in linkages between improved affect and cognition. Specifically, we explored the time-varying covariation between within-person change in global cognitive function and positive and negative affect. Well, it turns out on occasions when a given individual performed 1 unit lower on negative affect relative to their personal average, there was a corresponding improvement in their cognitive function. This suggests that cognitive performance scores within individuals are changing as a function of their negative affect, meaning that they're fluctuating together within individuals across time. So if a participant received a higher score for negative affect, indicating more negative affect or worsened mood relative to their own personal average, then their MMSE score was lower, and vice versa, a lower negative affect score subsequently resulted in a spike in cognitive performance. So in other words, these processes were fluctuating together across time, and this emphasis on coupled change provides us with a better understanding of how two processes synchronously vary within persons. So these findings imply that reducing comorbidities associated with dementia, such as negative affect, through participation in a lifestyle intervention may facilitate increases in psychosocial and cognitive function. And the reason why reduction of comorbidities might be effective has to do with the notion of resource competition. If negative affect, depressive symptoms, signs, etc. are mitigated, then this frees up neural resources, which might result in a bump in cognitive function, despite individuals having a progressive neuropathology. So what's next? Well, during the current pandemic, a shift towards virtual connection is particularly imperative for groups at greater risk of isolation. And social interaction has important and significant impacts on psychosocial well-being and quality of life, and as we saw previously, on cognitive functioning. So accordingly, and thanks to the amazing Voices in Motion team, the choir has transitioned to an online forum for individuals to continue partaking in the intervention and to maintain necessary social connections. Therefore, for my next proposed study, I will explore whether this virtual choir produces similar benefits to those already demonstrated in the in-person version. The primary objective is to demonstrate that reducing social isolation and depressive symptoms via virtual psychosocial engagements can reduce some of the many serious health risks linked to social isolation. A virtual socio-cognitive lifestyle intervention, such as Voices in Motion, has the potential to decrease isolation, maintain quality of relationships, and curtail depressive mood. And if a virtual socialization intervention can result in subsequent within-person increases in cognitive function, despite the irreparable neuropathological changes associated with dementia, then these results should be extended to wider populations. For example, those in residential care, in rural settings, those with severe depression. As the current healthcare system continues with social distancing and cautionary measures, specifically for high-risk populations, it's crucial to understand and potentially harness the power of modifiable risk factors such as negative affect and social isolation. So in concluding, just want to give a big thank you to Stuart for being an amazing supervisor, and a thank you to the incredible Voices in Motion team and the funders, the Institute on Aging and Lifelong Health, and a very large thank you to the PBC Health Benefits Society for their incredibly generous donation and I'm so grateful and appreciative. Thank you.
Thank you so much, Cindy. Excellent presentation, and so nice to see this continue virtually and to see more results coming in the future. Thank you. Our last awardee is Ashleigh Parker, and for the David Chunyan Lai Scholarship. And Jody Garlock will introduce.
Thank you. It's my pleasure to introduce Ashleigh Parker today. Ashleigh completed her master's degree in my lab, during which time she learned multiple neuroimaging analysis approaches very impressively. And this was all to better understand biomarkers for Alzheimer's disease. Ashleigh is continuing on this work as a PhD student in the clinical psychology program and specializing in neuropsychology. I've been so grateful to have the opportunity to work with Ashleigh. She brings energy and enthusiasm to her work, and she's established collaborations within my lab, within UVic, and all the way to Australia at the University of Melbourne. Ashleigh's current work focuses on establishing neuroimaging-based biomarkers for Alzheimer's disease, with a focus on people who have subjective cognitive decline. So thank you so much for this award, and welcome to Ashleigh.
Thank you so much, Jody, for that introduction. And I'm gonna test my luck at sharing my screen. Let's see if I can. Here. And has everyone seen my slides? Perfect. All right, well, yes, thank you everyone for being here, and thank you so much to the generous donors that have generously provided their support to graduate students at UVic. And as Jody was mentioning, my study for my doctoral research will be focusing on the early detection of Alzheimer's disease using MRI. So just before I jump into my study, I want to give some background into why it's important that I'm looking at why I'm looking at this group. So when we age, there's an increased risk of developing Alzheimer's disease, and Alzheimer's disease makes up approximately 70% of all dementia cases. The term dementia refers to a general decline in overall mental abilities, and it could be caused by a number of different conditions or diseases. Alzheimer's disease is characterized by memory impairment and underlying neuropathology, which includes beta amyloid plaques, neurofibrillary tangles, and neurodegeneration. There is a late clinical diagnosis where an individual is not usually diagnosed with Alzheimer's disease until later stages, where irreversible neurodegeneration has already taken place. Currently, there is no cure for Alzheimer's disease. Therefore, earlier identification of individuals who are at a higher risk of developing Alzheimer's disease is necessary. Here in this figure, it's showing us the trajectory of cognitive decline in normal aging, as well as the trajectory of cognitive decline in non-normal aging, such as what we would see in dementia. Cognitive decline is shown on the vertical axis, where lower points on this graph represent more significant levels of cognitive decline, while the horizontal shows time over years. Down on the bottom right is what we consider the most severe of cognitive decline, because someone in these phases would show levels of cognitive impairment that would interfere with their ability to carry out everyday abilities. To the left is mild cognitive impairment, also known as MCI. In this phase, the individual may be experiencing cognitive changes that are of concern to themselves and or to their loved ones. In MCI, one or more cognitive domains are significantly impaired, but their abilities to carry out daily activities is still relatively preserved. Although this is the first stage where detectable differences in cognition can be found, as on cognitive assessments, there is already irreversible damage done to the brain. So it makes sense for us to start looking a little bit earlier at this preclinical stage known as subjective cognitive decline. Here in this phase, the individual experiencing the decline may notice differences in their own cognition, but this decline would not be detectable on cognitive tests, meaning that someone in this phase could still perform within the normal range of scores on these assessments, even though they are noticing a problem in their own cognition. In 2014, a working group called the Subjective Cognitive Decline Initiative operationalized the term subjective cognitive decline, or SCD, aiming to reach a consensus of terminology and create a conceptual framework for researching SCD. Research criteria for SCD requires the individual to, 1, have a self-experienced decline in their cognitive capacity in comparison to their previously normal status, and 2, perform within normal limits on standardized cognitive tests that are adjusted for their age, gender, and level of education. Both of these criteria must be met for an individual to be considered to have SCD. An individual would not be considered to have SCD if they have been diagnosed with MCI, prodromal Alzheimer's disease, or dementia. They would also not be considered to have SCD if the reason for their decline in cognition could be better explained by a psychiatric or neurologic disease, such apart from Alzheimer's disease, or another medical disorder, a side effect from a medication, or from substance abuse. This working group also believes that in this preclinical stage of Alzheimer's disease, there's likely to be detectable biomarkers. And a biomarker is a biological characteristic that can be identified in vivo and is indicative of risk or presence of a disease. One way that we can measure biomarkers is through magnetic resonance imaging, or MRI. MRI is an ideal technique for biomarker detection because it is non-invasive, it does not expose the individual to any radiation, it is easily repeatable, widely available, and capable of producing high-resolution images of the body, including the brain, which is the area I'm particularly interested in. MRI is also capable of acquiring different types of data all on the same machine, such as structural MRI and functional MRI. And both of these types of analyses will be the focus of my doctoral research. So for my doctoral research, I will be utilizing participants from the Women's Healthy Aging Project, which is led by a neurologist who is Dr. Cassandra Serkey, who is co-supervising me for this project. And the Women's Healthy Aging Project has contributed to over 200 published research findings, but so far its MRI data has been underutilized since one requires certain knowledge to be able to analyze this data. For my project, the group that I'm looking at includes healthy controls as well as women with subjective cognitive decline. And we have a total sample of 170 women. And along with the MRI data, this data set includes cognitive data and questionnaires on so many different topics, lifestyle, relationships, and even has other data within their research, including hormone levels. So for my study, I'm most interested in investigating brain differences between healthy women and women with subjective cognitive decline. Specifically, I'm interested in looking at whether there are differences in brain tissue density between those with SCD and healthy controls. And using my master's research to inform this hypothesis, we hypothesize that those with subjective cognitive decline will not show differences in brain tissue density using voxel-based morphometry. So looking at gray matter changes in the brain. Another area of interest is determining whether there are differences in functional connectivity using resting state fMRI between healthy women and women with SCD. And again, using my master's research to inform this hypothesis, we hypothesize that women with SCD will show both increased and decreased functional connectivity in the default mode network using resting state fMRI. And to emphasize why I particularly find this area interesting is that I'm looking at these groups of individuals that are very similar to one another, except for one thing. So the women with SCD self-report that change in their own cognition, and the healthy controls do not. These groups both represent women who are in good health and both perform within normal limits on cognitive testing. The only difference is that self-report of decline in cognition. So I've conducted a preliminary analysis of this data with the voxel-based morphometry, looking at structural differences between women with SCD and healthy controls, and no difference between groups was found when using this approach. And this is exciting because it does line up with my previous research and our current hypothesis, and is also really exciting because when we look at these other metrics, including Fmri, perhaps we're able to see that there's differences in brain function before we see that structural atrophy and before irreversible neurodegeneration occurs. In the future, I will be examining the entire sample and as well include that functional connectivity analysis. And for the sake of time, I won't get too much into what else we're looking at, but we will also be looking at white matter analyses and a longitudinal analysis where there's a subset of women who were scanned in 2002 and in 2012. So we will have some access to longitudinal data, which is really exciting. And implications of this research is that my project really has the opportunity to represent a crucial step in characterizing subjective cognitive decline, which is a group recognized to be at an increased risk for developing Alzheimer's disease. Future study of biomarkers in SCD is imperative so that disease delaying intervention may be implemented at the earliest time point in order to reduce the devastating effects of this neurodegenerative disorder. Thank you so much, everyone, for listening to my presentation, and thank you again to the generous donors.
Ashleigh, thank you so much. Terrific presentation, and Jody, to you. Cheers to all the student awardees and just who gave excellent presentations. Nice to share what's going on here. We should do this more often. It's so good. We're going to move to our question and answer period, and then I understand there's going to be a couple of breakout rooms for some students to meet with their sponsors. But I just wanted to take the opportunity to again thank the sponsors for the many different awards that the Institute gives out every year. These are very important to support students and to support their research in important ways that allow them to acquire the supplies, the facilities, the research payments, whatever is needed to accomplish some of these really rather complex research projects. And I just really wanted to thank the sponsors for that, but also to the students and their supervisors, and also to Joanna and Leah for supporting the awards process throughout the year and for organizing this session. So with that, let's move on to some more open questions, answers, discussion. And Joanne, I understand you might have been keeping track of the chat, and maybe you could help organize this. Thank you.
Well, there's not much in the chat, and we're a small group, so maybe if people have questions, they can feel free to step forward and maybe identify whether it's a question for the group as a whole or for particular individuals. Well, if nobody has a question, I do for all of the students. Some of you mentioned having to pivot a little bit because of COVID. One of the questions I have, particularly since, but this applies to Maddie as well, particularly since many of you are in sight, is with some of the symptoms we're seeing with the COVID pandemic and some of the beliefs that those are going to be long-term consequences. Can each of you sort of comment from where you are in your current research about what you think might be things we should look for in the future, and not limited just to the aging population, because we've got so many, we've got the kids and the early adults also being affected by this. Let's start at the top. So maybe Maddie's, oh, thank you, Maddie, you're gonna jump in.
Thank you. That's a very good question, Joanne, because while I was listening to everyone, I was thinking about this common theme of the social component, or maybe because it's part of my interest, right? But I think COVID is teaching us and remind us the importance of social connection. So now we are finding alternatives to have that social connection, maybe through online or with apps. But it is important because it really has an effect, a real effect in our brain. So it is important to consider that. And I will say that also music. And when I was being part of this, I got a message on Instagram from a friend in Mexico and was telling me, Maddie, thank you for suggesting putting music to my grandma because she's so isolated. But now she's listening to music and I can see how her mood is changing and it's improving just because music. So I think what is happening now with COVID is that we need to come up with more creative solutions that maybe they have been there all the time, but now it's the time to really hand and use it, because we are limited with other options. But I will say, just to finalize, that music is powerful for the brain and social engagement as well, a lifestyle with social engagement. And we have to find alternatives with the COVID situation. Yeah, thank you.
Ashleigh.
Hi, this is more coming from maybe a clinical perspective, but we have discussed a bit in our program about how this might be like a clinical population that we'll have to think about working with in the future, people who have these long-standing cognitive impacts. And I haven't gotten much training in cognitive rehabilitation yet, but that's really something I'm hoping to focus on when I practice later. And this, just wanted to share that our program has really discussed that this is something we need to be looking out for at all ages later on, and how can we help those who are still having lasting cognitive impacts.
Mm-hmm. Jamie, did you want to jump in there?
Yes, I mean, I've been just so impressed with across the globe how many different ways in which people have been able to pivot and just connect with each other and learn and grow, and all the researchers who have been trying to sort of make sure that the populations that need it the most, if there are, like rural populations or people who might be more affected, and all of the governments who have been giving extra funding and things like that for people to make it through, with small businesses and all of those kinds of things. A lot of news has been surrounding, of course, my area, and how smell can often be impacted as one of the first indicators as well for COVID. And those people, of course, will also have long-standing potential impacts with extended issues with their sense of smell. So there's also potential impacts there for their well-being on a long-term basis, having COVID and then just having a long-term issue of their smell potentially not recovering. But there are olfactory training protocols that are going out now. So a lot of researchers have been really sort of pivoting and focusing on these areas, which has been so nice to see.
Colleen, did you want to comment?
Yeah, sure. So what the kind of global context in terms of the virus has had me thinking about is kind of, how COVID might be contributing to different developmental trajectories for individuals who might already have risk factors for something like dementia. Like, how might the pandemic in the future, like people who've had the virus, how would that interplay into that? But then also listening to the different research talks today on ways to kind of is neural resource kind of enrichment and difficulties with music and with socialization, it's kind of inspiring to think that maybe that might be an opportunity to counteract that, those possible negative long-term effects. So I think it's just a matter of staying tuned and continuing on with all the different research that everyone has been talking about today, and kind of tracking that and keeping that in mind for those individuals who have had the virus. So.
Cindy, over to you. Did you have any comments?
Yeah, one large aspect that I've been thinking about lately is kind of the difference between social support and actually feeling supported, and how many of these target populations maybe have these social support virtual interventions available to them, but is that really helping them feel supported and safe, and are they actually utilizing these programs available to them? So I think that's going to be a very interesting avenue of upcoming research as well, similar to the Voices Choir. Are the benefits similar to the in-person version? And then what are the impacts on cognitive health based on loneliness and isolation?
Hey, and over to you, Nathan, for the last this particular question.
Yeah, so all very good points that have been mentioned so far. I think I'm forever the optimist, so I'd like to end on a positive note that yes, I think there are definitely some issues with mental health that could emerge in response to the pandemic, but what's really amazed me is how people have tried to adapt in different ways, and kind of tying it back to cognitive reserve. I've seen so many people trying to learn a new musical instrument, or are reading a lot more, or have taken up all these different hobbies that hopefully they keep up, and these are things that could have lasting impacts on their cognitive health throughout the lifespan. So I don't think it's all necessarily doom and gloom related to COVID.
So I think of cognitive reserve as something that's being built up over a lifetime, and you raise a good point for the senior, the current senior group. What about the folks in their 20s, 30s, 40s that have had COVID, or maybe even some of the symptoms of COVID, and whether or not they're likely to be able to develop some of the cognitive reserve that might be required 10, 20, 30 years down the road?
Yeah, well, I think we have similar views about cognitive reserve. I see it as something that it's not just something that you do later in life or something that's done early in life. It's something that's reflecting your kind of cumulative engagement throughout your lifespan in terms of mental stimulation. And I mentioned that education is the big one that a lot of researchers in cognitive reserve tend to focus on, and that's something that's typically happening in the early 20s, oftentimes. So I would say it's more about kind of setting up the kind of lifestyle that you'd want down the road, developing these kind of habits, because it's so important to start early. If you're going to be an avid reader, for example, kind of starting early, developing that as a habit that you integrate with your all the responsibilities in your life. I think that's where it would have lasting impacts and hopefully benefits down the road.
Well, I think it's going to be interesting times for all of us as we experience the fallout of this. Margaret or Kathy, did either of you have questions either for the students that received your awards or for the group as a whole?
I don't have any initial questions at all. I'm just so impressed with the kind of things that are being done by all of the students today that received awards. I think it really gives us all hope, and for the future when people are looking into these kinds of things at the level that I've seen here today. So I just want to congratulate everyone here.
Thank you. Kathy, did you have any questions or want to comment?
No, I'm in the same position as Margaret. I think that the hope that I see in this. And Nathan, I'm with you. I tend to look at things at the positive aspect as well, and that's where I go first. And so what I see is a whole lot of hope for my future as an aging British Columbian, and certainly support for our members. And at PBC, we have a high volume of aging members there as well. And I think that this is tremendous research, and congratulations to all of you. Wonderfully done.
And Moses, this puts you in a different kind of spot, but just with the range of projects that the students have talked about, is there any comments from your end in terms of the development perspective?
Hello. Well, thank you. First of all, great to hear all those very interesting research projects that you're working on. And I think, I don't know, Kathy, like you said, that you're pleased to hear some of the research. I know Chris Ferguson wanted to be here, but he just messaged me a little while ago just saying that he had a conflict and couldn't make it. But I think hearing what you guys are doing kind of inspires me to try and find more funding to help with your research and to help with your work. And I'm glad that Kathy had a chance to listen. And I know that the Pacific Blue Cross Benefit Society is very interested in the work, and I think we're still in conversation to see if we could take some of this funding a little bit further, more beyond year 2. So I'm glad to have heard what you're working on, and looking forward to hopefully seeing that video. I think, Joanne, you mentioned that it's being recorded, and Chris had an interest to really hear from the students and see some of the work that's being done. So,
thank you. Did anybody else have any questions or comments? Otherwise, I'm going to jump in again.
Joanne, do you have a question?
Why, thank you for asking, Scott. It struck me actually as we were going through about the various aspects of the cognitive decline. Does anybody know what cognitive decline looks like in specialized populations? For example, our street people, or our folks that are in the various gender populations, and how that is the same or different, or would we expect it to be the same or different? Does it occur at a different age? Does it look different when we have some other things happening? So over to anybody that wants to grab it, including the faculty, because I don't know the answer to the question. I would expect that with our street population that it might kick in sooner. But I'm thinking with all the socioeconomic consequences and the intersectionality with mental health, drug and alcohol abuse, that the actual brain damage due to cognitive decline as a result of faster aging may not be picked up the same and may not look the same. Because I was thinking, this is actually prompted by your work, Nathan, on resilience, and just wondering if there's other things that can be done with some of those special populations to help build the resilience and therefore impact the cognitive functioning. I don't know. Any comments, thoughts? Does anybody know the literature in this area?
I don't have specific knowledge of those marginalized populations off the top of my head, but something that I can speak to is that there are factors that go into why people experience subjective cognitive decline, and potential factors could include types of medications, if they're experiencing anxiety, if they're under extreme stress, depression, fatigue. So all of those different things can impact someone's ability to think properly, or for them to experience that subjective decline in their cognitive abilities to what they remembered before. And we also know of normal aging cognitive related declines. So when we're speaking about people who are afraid of getting dementia, it's not from forgetting where your keys were once, or if you can't remember someone's name you just met a couple times. It goes farther than that for not being able to recall events or recent things that have happened to you or full conversations. But these little things that we can pick up on are sometimes more representative of, that normal aging decline in some of our cognitive abilities. But I could certainly see that it would be exacerbated in marginalized population who are under extreme stress. They experience like microaggressions every day, worrying about, homelessness, where are they going to get their next meal? That could definitely play into all of that stress and fatigue and perpetuate those problems.
Oh, and from Jamie's work, the impact, if you're losing your sense of smell and you're on the street, very different than if you're in safer environments, right? And I think for Cindy and Maddie, I don't think it's ever been explored within this perspective, but Louise Rose, a local jazz musician, has for many years run a choir to support the folks at our place. And I don't know whether anybody's ever looked at even the psychosocial benefits of that. That's part of why I think Louise has done it, but I don't think anybody's ever formally researched that. Maddie, did you want to say something?
I know her personally because we go to the same church. Her work and her music is awesome. One of the limitations, and I didn't mention this, but one of the limitations that I pointed out for Voices in Motion is that the sample is not very heterogeneous. It's actually very homogeneous. So we didn't have much opportunity there to explore like those themes around intersectionality. But it makes me think also about the equity component, right? Because it is so important. Just when Nathan was talking about the cognitive reserve and the lifestyle, he put two columns, and in one column, talking about lifestyle, it was saying like poor education, mood disturbance, poor nutrition, alcohol and drug abuse, and poor health can drive to healthy cognitive reserve, but in a negative way. So all of those that I just mentioned are part of the challenges for some marginalized communities and people, for sure. So just thinking about that, but I haven't done research on that, and I don't know about how is the sample for the others groups here, but I haven't found many. I know there is one choir in San Francisco that is also with like black community, and the Latino community are part of it. But I don't know for others here. Yeah, thank you.
Did anybody else want to comment? All right, get a different group, and probably again, and not an answerable question, is just what is the incidence of dementia in people, for example, like professional musicians? That goes to Cindy's, some of Cindy's work. I'd be curious to know whether or not there is, whether it's better or worse in quotes than the normal population.
Yeah, this actually relates a lot to Nathan and cognitive reserve as well. And like he was saying, the more kind of reserve you have, then perhaps the later symptoms start to arise as well. And in regard to, shout out to Stuart, there's a selection optimization compensation model, which is basically it's a way of, for older individuals who are experiencing kind of like slower declines, functional declines, cognitive declines, how are they compensating so that they can still perform all of these activities, and their outlooks on being able to maybe just change the way that they approach some of these musical talents, and then still being able to maintain you know, a lot of these functions and that kind of thing. In regards to the prevalence rates, I can't give you that off the top of my head, but yeah, I believe that musical ability and that kind of talent and focus contributes to cognitive reserve in the long run.
Yeah, it just struck me that when you guys were talking, that you would expect with professional musicians, I would expect professional musicians, that either any brain damage that was there would be detected later and steeper, or there would be preserved functions. So maybe while other areas are being lost, that musical strength can remain. Just thinking of folks like Bob Dylan, for example, with the significant head injury, but how he's still able to function on some level, just not across the board.
Absolutely, yeah, and like we were saying, in Alzheimer's populations, music memory system is relatively spared because it's very specific, and it's less tied to like episodic memory and brain systems that are more initially impaired in the disease process.
Maddie, did you want to say something?
Yeah, this conversation remind me an article that I read the other day, and it differentiates that when you are singing in a choir to enjoy is different than when you are singing in a choir professionally, because then you have the component of a stress. Like if it's your job, it's different that if you have this disposition to enjoy as a recreative activity. But also I want to mention that maybe you are from BC and maybe you know the band Spirit of the West, do you know? And I heard anecdotally the other day, a friend told me that one of the singers, he used to live here in Vancouver Island in Victoria, and he got dementia. And he was in his garden, they used to have little concert with friends. And he was not in an early stage, he was in a more advanced stage, but he liked to play and he can remind all the songs and share with friends in the garden here in Victoria. So it was kind of like a good anecdote that a friend was sharing with me the other day about this band from the 90s.
Okay, well, anybody else have any questions?
I have a quick comment, Maddie. There's a great documentary called Spirit Unforgettable that you can actually watch, almost a 2-hour story that chronicles the journey between him and actually his wife, Jill Daum, who's a playwright and has actually co-wrote and directed or at least contributed to a recent play on the topic of dementia. Yeah, and John Mann was his name. He had a presenilin mutation, so he had an early onset variant. He was young when he was affected by it. Yeah, and I just echo Cindy's point. I think, Joanne, per your question, yeah, there's great literature. Certainly Jody will be familiar with it. Russ Poldrack, 2001, Proceduralization of Skill. Rather than forcing people with a deficit to do things that draw on impaired systems, stack the deck in their favor and give them an opportunity to engage them in things that put them on the equal footing. And choral singing is one of those things because it can draw on, striatum and other things that are fairly well preserved, even in more advanced stages of the disease process. And yeah, there's a great YouTube clip of a gentleman who was a lawyer. He was in his 70s, pretty advanced dementia, experiencing all of the typical symptoms, but he learned piano when he was very young. He wasn't a professional musician by any stretch of the imagination, but he just had this profound quote where he said, yeah, I just go and play because music is a place I can go where memory doesn't have any effect on me. And that's the old adage, like riding a bike. It's yeah, proceduralization of skill.
Just a second. I would echo what others said. I think this was a great opportunity to learn about your research. Thank you so much to all of the students for presenting, and thank you to the supervisors for introducing your students and also attending. It means a lot to the Institute for your participation. Scott, back to you.
I would just add my thanks to what Joanne just said. Thank you all for being a big part of this, and we're just so delighted to get together with you in these days. So wishing you all well, stay well.